Microscopy, the discovery and study of cell, progressed with the invention of microscopes. Microscopy is the technique used to view objects that cannot be seen by the naked eye. The microscope that you use in the laboratory are light microscope. Light microscope use visible light to illuminate specimens into a two-lens system. The two lenses present in a light microscope are the ocular lenses in the eyepiece and the objective lens located in the revolving nosepiece. As the light microscope uses two lenses, so it is known as compound microscope. Working principle: the light microscope light source illuminate the object. The first image of the object is formed by the objective lens. It is a real, inverted, and magnified image. The image formed by the objective function as the object for eyepiece. The eyepiece produces the final virtual and magnified image. In this way, the final image produced is inverted with respect to the object. Magnification. The magnification is the capacity of an optical instrument to increase the size of an object than its original size. The objects which cannot be seen by naked eye can also be observed by increasing magnification. Different lenses have different magnification powers, which are represented by the letter X. That means that the number of times of that original size. Therefore, a lens of 10x magnification can be increases the size of an object of 1 micrometer to 100 micrometer. The total magnification can be calculated by multiplying the objective lens value by ocular lens value. Maximum magnification of a light microscope is 1500x. Using a 100x objective and 15x ocular lens, an electron microscope can magnify specimen between 1 and 50 million times depending on which type is used. Resolution. It is a measure of microscope's ability to distinguish between two points which are closer on an object. If a microscope can clearly distinguish between two points, it has a good or high resolution. The resolution of naked eye is 0.0 millimeter. This resolution cannot be increased by increasing magnification. The resolving power of light microscope is 250 nanometer The resolving power of an electron microscope is 0.2 nanometer. Difference. The difference between magnification and resolution is that magnification is the ability to make small objects seen larger by using optical instruments. For example, microscope. Resolution is the ability to distinguish between two objects from each other. Electron microscopy. The principle of electron microscopy. It uses the beam of electrons to visualize the specimen and has a high resolution. The working principle of electron microscope. The beam of electron are produced from the electron gun is focused on the specimen through two sets of condenser lens. An accelerating potential is applied between the filament and anode to move the electron downward. The specimen to be observed is kept as thin section of 20 to 100 nanometer on specimen holder. The beam of electron goes through specimen are the denser region scatter more electron on the screen than the light regions. The beam of electron scattering from specimen passes through the objective lens to create magnified image which is further magnified by the ocular lens.